Okay. So, recap. Watson and Crick work out that DNA has this shape. It's shaped like a double helix, two polymers of nucleotides, two strands, each one twisted about the other, looking a little like a braid or a rope. In this diagram, the sugars and phosphates have just been replaced with this ribbon, but you can see how the bases are these spiky things that are sticking out and bonding with each other and forming these hydrogen bonds that make up the rungs of this twisted ladder shape. So it's not really shown, but right there, there's hydrogen bonds, and there's some, and there's some, and there's some, and they make up the rungs of this twisted ladder. And you may be wondering why on earth you should care. So we have this tiny, tiny, tiny thing in our cells that's shaped like a twisted ladder. Okay, DNA is a polymer, or more accurately, two polymers of nucleotides wrapped around each other, with the two polymers held together by hydrogen bonds between the bases. And each nucleotide consists of subparts: a phosphate group, which is an atom of phosphorus with some oxygen stuck onto it. That's the phosphate. A simple sugar, that's a ring of carbons, hydrogens, and oxygens. And in this case, okay, I really can't write on this thing. Sugar. In this case, the sugar is a type called deoxyribose. And then one of four possible bases, which could be adenine, thymine, guanine, or cytosine. Okay, that's just a recap of last Tuesday. We've already talked about how the double helix structure allows DNA to duplicate, how each half of the DNA helix separates from the other half, and then new nucleotides come in and assemble themselves in the same order as the order of nucleotides on the other strand. That matters because there is something crucially important about the order the nucleotides are in. When DNA was first discovered, people thought, okay, maybe it's just, some kind of repeating pattern like ACGT, ACGT, ACGT or something, or maybe it's A, C, 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 or, something like that. It turns out it's a lot more complicated than that. The order isn't random. The order actually carries information. And it's been possible, and since the early 90s, it's been relatively simple, if you've got the right equipment, to take a piece of DNA and literally read off the order of nucleotides on a DNA strand. So, for example, this is a piece of human DNA, and I don't remember off the bat what it is, but there is some piece of your chromosomes where on one of the strands of DNA, the nucleotides go C, A, G, C, T, G, 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 T, A, A, G, 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 C, G, G, A, T, T, A, T, T, C, A, T, A, T, A, A, T, T, G, T, T, A. Hope you're writing all that down. I'm kidding. You don't actually have to memorize this. So one strand has that, and then of course the complementary strand would go G, T, C, G, A, C, 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 and so on. If you know one of the strands, you can figure out the order on the other. Although DNA is actually different for different people. And in fact, if this was DNA from a Canadian, it would say A, an awful lot. Sorry. And then if it was DNA from a Spanish speaker, it would say C, 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 an awful lot. Right. You're not obliged to laugh. Okay. The reason this matters is that order contains information, but to know what we do with the information, we have to look at a second class of large molecules called proteins. And this is one that you've probably heard of. It's certainly important if you pay attention to diet and nutrition. You're supposed to eat a certain amount of protein. You know, it's good for your muscles and things like that. Proteins are one of the 4 classes of big molecules, and we talked about those way back at the start. And they do an absolutely ridiculous number of things in a cell. Like nucleotides, proteins are, or like DNA, proteins are polymers, but they're polymers of a type of subunit called amino acids. And there's 4 bases that make up DNA. They can be put together in different orders. There are 20 different amino acids that are used to make proteins. There's slight variation in what those are in different organisms. For our purposes, it's 20. You put the amino acids together in an order, and that determines the shape of a protein molecule. And as I've talked about with DNA, it's true here too. The shape of the protein molecule determines what the protein does. The link is that DNA carries information coded in the order of nucleotides that tells a cell how to put the amino acids together in the order that will give it a protein of the right shape. DNA stores information. The information is used to assemble proteins. And if you assemble proteins correctly, they'll do things in the cell. And we'll look at what those things are. May not be until next Tuesday, but we'll cover as much as we can today. So an amino acid has parts, just like a nucleotide has parts. Every amino acid has, well, it's got a little central carbon atom with a hydrogen stuck to it right there. I kind of left that out. But sticking to that central carbon, it has an atom of nitrogen with a few hydrogens on it, and that's called the amino group. On the other side, it's got a carbon with 2 oxygens and a hydrogen stuck to it. That grouping of atoms is called a carboxyl group. And then the third thing that's sticking to that central carbon could be anything from a single atom of hydrogen to a big chain of carbons, hydrogens, and nitrogens, to a smaller chain, to a ring, could be almost anything. And we call that the R group right there. So there's the amino group, there's the carboxyl group, and then different amino acids are defined by having different R groups. They've all got amino and carboxyl groups, but depending on what the amino acid is, the R group could be quite a lot of things. And of course, amino acids are very important in the diet of pirates because they need R groups. Thank you. There's actually many thousands of amino acids. This is one that I don't know how well it works or not. It's not really my field. Some of you may have more experience than I do, but ornithine is an example of an amino acid that is not used in assembling proteins, but there's other roles in metabolism. And it's sometimes taken by people who are into fitness. Off the top of my head, I'm not quite sure what it does. And I'm not sure whether it works, but it is sold as a supplement. And I show you this because it's an example of an amino acid that is not one of the 20. There's other amino acids that your body might use in various roles in metabolism that aren't part of the 20. There's many thousands of possible amino acids, each with a different R group, but there's only 20 that are used to assemble proteins. Again, slight variation in different groups of organisms, but for the Cliff's Notes version I'm giving you here, there's 20. And incidentally, you might have heard of this if you follow up on nutrition. 11 of those 20 your cells can make if you don't get enough in your diet, but there's 9 that your cells can't make, and those have to be present in the diet. We call these the essential amino acids. Actually, for infants, I think there's 10 or 11 that are essential. Newborns can't make some amino acids, but they're able to make them when they grow up, which is why baby formula has to be formulated so carefully. Anyway, food that provides enough of all 9 amino acids, we say, is a complete protein. So beans and rice, for example, is a complete protein that contains all 9 of the essential amino acids, and any that are missing, your body can make. If you're not eating a complete protein, you can end up with various deficiency diseases. I think if you live only on corn, you can end up with a deficiency disease. But if you mix your corn and your beans, you'll be fine. So that's what essential amino acids are. And amino acids have these 3 basic parts: the amino group and the carboxyl group, which all amino acids have. And then what makes different amino acids different is they've all got different R groups. And those things I'm circling right there, those are the R groups. Okay, I'm not printing very clearly this morning, but yeah, those are R groups. Okay, I won't repeat my joke about pirates, but I will tell you I heard that on the pirate radio network. It's called NPR. Right. Okay, right. Do not remotely make an attempt to memorize these. This just happens to be the formulas. Please mute somebody. Somebody, I'm getting music that, yeah, thank you. All right. These are some of the 20. Up here, you've got, that's the amino group and the carboxyl group up there, and the shaded part is the R group. Alanine, it's just, the R is just a carbon and 3 hydrogens. Phenylalanine, it's a carbon, a hydrogen, and then a ring of carbons. Tryptophan has this big fancy double ring. Methionine's actually got a sulfur in it. That's the S in schnapps. You need sulfur for certain amino acids. There's some more. And there's yet more. And again, you do not need to memorize those, but the R group in all cases is the shaded part. When you get 2 amino acids together and they like each other very much, they'll form a bond between the amino group of one acid and the carboxyl of another. In red on the left, we have an amino acid that happens to be called aspartic acid. And I've drawn it so that every atom is shown and represented by a letter. On the right in blue, we have the amino acid phenylalanine. There's the carboxyl group of aspartic acid, and it gets right next to the amino group of the phenylalanine. And when you put a carboxyl next to an amino, the aspartic acid carboxyl forms a bond with the amino group on the phenylalanine. This oxygen and hydrogen falls off. This hydrogen falls off, and they actually come together and form a molecule of 2 hydrogens and 1 oxygen, otherwise known as H2O, aka water. You actually make a molecule of water every time your cell does this. And then a new bond forms between this carbon over here and this nitrogen over there. And again, we can pretend for our purposes that chemical bonds are like little sticks. If you really want to know what they are, you can go bother the chemistry department. For here, we're not really going to worry about what is making these bonds happen. Not that it's not important, but we've got to finish the course in a finite amount of time. And that creates what we call a peptide bond. Now, peptide bond is a bond between the carboxyl on one amino acid and the amino group on the other. And here's the thing, here's a carboxyl right here. This carboxyl, in theory, could form a peptide bond with the amino group of another amino acid. And this amino right here could form a peptide bond with the carboxyl of the next amino acid. So you can link these together in chains indefinitely long. The largest I can think of is over 1,000 amino acids. I'm not sure what the actual maximum possible is, but the point is that you could stick these together because each amino acid has an exposed groups that can form peptide bonds with more amino acids. You know, just like every train car has a latch where you can attach another train car. Incidentally, if you'd stopped there and stuck a couple more atoms on one end and replaced them with this right here, a group of carbons and hydrogens, you would have a chemical called aspartame, better known as NutraSweet, one of the best known no-calorie sweeteners, the active ingredient in Diet Coke and a lot of other soft drinks and things. Yeah, so that's a nice example. Aspartame is basically 2 amino acids stuck together with 1 peptide bond. Now you know. But remember, we don't have to stop there. In theory, we could take an aspartame molecule and start sticking more amino acids on and forming a chain, one after the other, for, again, I think the record is over 1,000. You know, take it as, you know, for practical purposes, we could make that chain as long as we wanted to. To describe a protein, you can describe it on several different levels. And the lowest is you can simply describe the order in which you put the amino acids together, and we call that primary structure. It would be kind of like describing a poem by starting by describing the order in which you put the letters. THEREWASAMANFROMNANTUCKET, and so on, right? What happens when you do this, when a cell puts amino acids together in an order, they don't stay in a straight chain. The amino acid chain begins folding because some of the atoms in it have electrical charges, and those electrical charges can attract or repel each other. Similar to how electrical charges on your hair on a really dry day can attract or repel each other and leave you with, you know, flyaway hair that sticks to your sweater or your comb or something like that. So the amino acid chain begins to stick to itself, and areas of that chain will start folding or twisting in ways that we call secondary structure. That folded chain starts folding again, and that's caused by electrical attraction or repulsion between different R groups, and we call that tertiary structure. And then, as if that's not enough, you very often take several separate chains of amino acids. Each one of them folds into a shape determined by the secondary and tertiary structure. And then those folded chains all come together to make an even larger unit. Several separate chains of amino acids will fit together into one molecule, and we call that the protein's quaternary structure. So that's how we describe proteins, as having structure on different levels. It would be a little bit like, okay, let's take the UCA campus. The primary structure might be the list of bricks. The secondary structure might be the ways that the bricks are assembled. The tertiary structure might be the floor plans of the buildings, and the quaternary would be the plan of the whole campus. I'm not sure that works. Maybe don't pay too much attention to that metaphor. Let me try to explain it more directly. Primary structure is just the order in which you stick amino acids together one after the other. In this particular model, carbon atoms are represented as green sticks, and oxygens are represented by red sticks. So that's a carboxyl group, and that's the amino group of one amino acid. And that forms a bond with, there's the amino group and carboxyl group of another amino acid. There's the amino and carboxyl of yet another amino acid, and so on. So that's a model of what just a simple polymer of amino acids would look like if atoms look like sticks. We usually call just one polymer of amino acids a polypeptide, or just a peptide for short. So that's one polypeptide right there. And here's the primary structure of the human protein, insulin. You start with the amino acid methionine, and then there's an alanine, and then comes a leucine, and then tryptophan, and then another methionine and an arginine, and then two leucines and a proline and another two leucines, an alanine and two leucines, an alanine and a leucine and a tryptophan and a glycine and proline. And this is boring as hell. But even though you certainly don't have to memorize this, it ultimately matters, because cells in an organ called your pancreas assemble amino acids in this precise order to make a protein called insulin. If they don't assemble these amino acids in exactly the right order, then they don't make insulin, or the insulin they make doesn't work, and you have a condition called diabetes, of which you might have heard. Insulin is a hormone. It's you could think of it as a messenger that goes to your cells and basically says, hey, we've just eaten. There's lots of sugar in the bloodstream. All of you cells come absorb that sugar and use it to run your own metabolism. If your body can't do that for any reason, there's all kinds of unfortunate effects. You have insulin dependent diabetes, and it's all dependent on whether those cells in your pancreas can assemble the right amino acids in exactly the right order, the order that I'm showing you. So reading off the list of amino acids is boring as hell, but if your cells couldn't do it, you would have a severe illness. Multiply that by over 20,000, and that's what proteins do, because you make something like, oh, it's more than that, tens of thousands of different proteins. Okay. Primary structure is just the order that you stick amino acids in. Secondary structure is what happens when atoms in amino and carboxyl groups attract each other and cause the chain of amino acids to bend or fold. When a chain of amino acids naturally kinks itself into a spiral, we call that an alpha helix. Not the same as the double helix. Do not get double helixes confused with alpha helixes. A double helix is a DNA structure. An alpha helix is a protein structure. So a part of that chain might kink up into an alpha helix. Another part of that chain might actually kind of zigzag and fold back and forth into a structure that, if you could see it in 3 dimensions, would look kind of like a folded sheet. And that's called a beta sheet. You'll sometimes see it called a beta pleated sheet. So these affect regions of that chain of amino acids. So a straight chain of amino acids might look, just kind of like, one after the other might be straight if we could straighten it out. But when secondary structure comes into play, part of that chain might kink up and form a helix, and then part of it might not form much of anything. And then another part is going to zigzag and form a beta sheet, and then another part might not do anything, and there we might have another helix. The helixes and the sheets are these regions of a chain of amino acids. So secondary structure affects parts of that chain. Tertiary structure affects the whole thing. Attractions and repulsions between R groups cause the entire chain of amino acids to fold up. This is a protein that's found in muscles. It's important in transporting oxygen within your muscles. It's called myoglobin. In nature, it is kind of pinkish. The pink juice of raw meat isn't blood, because the blood has been drained, or at least it's supposed to be. But the pinkish juice, like inside a rare steak or something like that, is mostly myoglobin. And this particular protein happens to consist of several alpha helices. They've been diagrammed here. You're not seeing individual atoms here. We've kind of gone step back a level. And so these things that look like telephone cords, not that any of you post-millennials know what those are, get off my lawn. Those things that look like coiled telephone cords are alpha helices, and there's several of these, and that's secondary structure. And then the entire thing has wrapped itself up in a bundle, and that's tertiary structure. So this particular protein just happens to consist of many alpha helices bundled up with each other. This is one from cells in the intestine that happens to consist of several beta sheets that are kind of wrapped around each other. But most proteins consist of both. This happens to be called triose isomerase. For our purposes, we don't need to worry about what it does. But in red, each one of those red coily things is an alpha helix. And each of these green ribbony things is a beta sheet. And the purple ribbony parts are areas of the protein that are not alpha helixes or beta sheets. Not all of a protein has to, awesome question. Okay, not all of a protein has to be one or the other. How do we know proteins have this shape? Well, we know that they have this shape because of a lot of X-ray crystallography. Remember, that was the technique that Ross Franklin used to work out the shape of the DNA double helix. You can do the same thing with proteins, and we've gotten much better and better at doing that. Now, the goal that everybody has, or not everyone in the field, is it would be really awesome if we could just take the order of amino acids in a protein and calculate what shape the protein will have. That's turned out to be an incredibly difficult problem I met somebody back when I was a junior in an undergrad, back when I was your age, back in like 1989. Oh God, that's depressing. Who was working on the problem. And many years later, I guess it was about 5 years ago, there was actually a student when I was teaching this class one summer, who was actually a computer science major, and his major professor was working on this problem. It's turning out to be extremely difficult to compute what shape a protein would have if all you know is the amino acid order. It would be an awesome thing to be able to do because proteins do so many things. And it would, we could engineer proteins to have shapes to do anything we wanted them to, which is going to lead to things like new drugs. But we can't do it yet. It's a very knotty problem in math and computer science. So the way we know that proteins have these shapes is basically by doing an awful lot of X-ray crystallography. Here's some more examples. In these pictures, the alpha helices have been coded in blue, beta sheets are in red, and then the gray stuff that looks like wire, that represents parts of the amino acid chain that are neither alpha helix nor beta sheet. So yeah, that's just kind of eye candy. Finally, quaternary structure is what you have to talk about when you have several polypeptides, each of which has its own primary, secondary, and tertiary structure joining together to make a unit. Your cells make a protein that transports oxygen called hemoglobin, and your cells actually make two different types. They're called alpha globin and beta globin. We don't need to worry about the difference. But for them to function, two alphas and two betas have to come together and stick to each other and form a, whoops, did not mean to skip that, and form a unit called hemoglobin. And hemoglobin consists of two alpha globins and two beta globins joined together. Each one of those alpha or beta globins is a separate chain of amino acids that has its own primary, secondary, and tertiary structure. And by the way, hemoglobin is what makes your blood red. It binds with oxygen and transports it. So it's hemoglobin in your red blood cells that is carrying oxygen, even as we speak, from your lungs to every part of your body that needs it. And if your cells could not put together hemoglobins in exactly the right way, you could end up with a disorder like thalassemia or sickle cell anemia, or possibly something even worse. Right? Your well-being depends on your cells being able to do all this. This is a protein. It's a really important one, but for right now, we don't really have to worry about what it does. But it happens to consist of seven separate polypeptides. 6 of them are shaped kind of like wedges, and they fit together like this, seen from the top. They fit together kind of like those wedges in a Trivial Pursuit game, if you've ever played that. And they fit together around a 7th that actually forms this kind of central stalk. So the whole thing looks kind of like a mushroom. And so that's quaternary structure for you. This protein actually generates an awful lot of the metabolic energy that you use to run your metabolism. You could not live a second without it. And it consists of 7 separate polypeptides that all have to fit together into this kind of mushroom shape. So there. Oh, here's an example you can taste. Wheat contains proteins that are called gliadins and glutenins. And when you get a bunch of gliadin and glutenin proteins fitting together, they make up something called gluten. You may have heard of gluten because some people react rather badly to gluten. My wife, for example, her skin breaks out and her digestive system goes bad. I'll stop there. But she gets sick when she consumes gluten. Gluten is just the collective mixture of proteins called gliadins. I think this is a gliadin, and proteins called glutenins. And here in this picture, the alpha helices are kind of copper colored, the beta sheets are blue, and the parts that are not a helix or a sheet are purple. Okay, suppose you take a variety of wheat that's been bred to have lots of protein in it, and you mix it with water, and then you start kneading it, squashing the dough and rolling it out and squashing it and punching it and squashing it. That causes the gliadins and the glutenins to assemble into a giant quaternary structure. And if you knead it enough, that giant quaternary structure will fill the entire dough. And that giant quaternary structure is very stretchy. Pizza is made from high protein varieties of wheat. And it's all those proteins that are self-assembling into that ginormous quaternary structure that makes pizza dough so stretchy that you can throw it and spin it like this guy is doing. I assume you have seen this before. People will take pizza dough, flatten it out, and then throw it in the air and spin it around to make it round and flat. That's possible because they're using a wheat variety with lots of protein, and the proteins self-assemble into this big quaternary structure that holds the dough together. Now, on the other hand, flour for cakes and pastries and pie crusts requires low protein varieties of wheat. And when there's not much protein, and when it's not made into a dough that you need, that you punch and squish, that giant quaternary structure doesn't form. And the end result is a baked good that is delicate and crumbly instead of being chewy, right? Nobody wants a birthday cake that's chewy. Nobody wants a chewy pie crust, right? Or chewy biscuits. You're going for light and flaky and crumbly here. So the flour that you'd want to use for making pizza crust is much higher in protein than the flour that you would use for baking cakes. And if you've ever paid attention to this in the supermarket, or if you bake yourself, you can actually buy pizza flour or cake flour. All-purpose flour is kind of in the middle, and it's okay for a wide range of uses. But if you were like a serious pastry chef, you would want to get low protein pastry flour. Or if you were serious about making pizza, you'd want to get a high protein pizza flour. And the difference has to do with the amounts of proteins in the wheat. In our household, I always bake with gluten-free flour that usually has non-gluten proteins added. Sometimes it's whey. That's proteins from whey is what's left over after you take after you make cheese. They might have egg or legume proteins like bean or pea flour added to imitate the effects of gluten. I've gotten increasingly good, if I say so myself, at baking gluten-free bread, but it still always comes out more crumbly and less stretchy than bread with actual gluten in it. And this is true for the commercial brands as well. Gluten-free bread is never going to be quite as stretchy as, glutinous bread. So it goes. Oh yeah, by the way, I should not be making commercial endorsements here, but Bob's Red Mill gluten-free 1-to-1 is what I always use, and it always comes out pretty well. The Bob's Red Mill Company did not pay me to say that. So, this is a lot. Proteins have these complex shapes, and we can describe the shapes on 4 different structural levels. We can talk about primary structure, that's the order of the amino acids. Secondary structure, we're talking about parts of that chain of amino acids that fold up into helixes or sheets. Tertiary structure is how the entire thing folds up together. And quaternary structure is how several different chains of amino acids may fit together to make a structure that's even larger. Who cares? You make an absolutely ridiculous number of types of protein. Probably, I said 20,000 earlier, it may be closer to 30,000. I can't remember off the bat what the current estimate is, but you make tens of thousands of different proteins. And the proteins do a ridiculously large number of things. The collagen that is holding your cells together, anybody remember the extracellular matrix? That's a protein. The forms of collagen that form these strong fibers that bundle together to make your tendons, like the Achilles tendon at the back of your heel, or the big fibrous trapezius tendon at the back of your neck. That's protein, that collagen. Shoot, your muscles contract because of about 50 proteins working together. Your hair and your fingernails are made of protein. Proteins carry oxygen around. Proteins form the matrix for your bones. Proteins do crazy stuff. And you could not live if you couldn't make all of those proteins with exactly the right shape, because the function of a protein depends on the shape that it's got. And incidentally, you can change the shape of protein molecules by applying heat or acid or sometimes other chemical treatments. And when we do that, that's called denaturing the protein. If you want a really simple example, fry an egg sometime. Egg white is made up mostly of a protein called albumin. And when it comes out of the egg, it is, liquid and gooey and transparent. If you apply heat in a frying pan, that transparent gooey protein starts turning solid and opaque. It starts turning white instead of transparent. You're not breaking the protein down unless you burn the egg, but if you fry it or hard boil it, you change the shape of the proteins, and that changes the entire substance into the solid white substance that is cooked egg white. Oh, if you want another example, your hair is made up of proteins, and the texture of your hair, whether it's curly or wavy or straight, depends on the shape of the individual hairs. And the shape of the hairs depends on the shape of the keratin proteins that make them up. And you can chemically treat your hair with heat and chemicals so that the proteins take on a different shape, and that changes the shape of your individual hairs, and that changes the entire shape of your hairdo. This is, of course, getting a perm. If you've ever had a permanent wave, what they're doing is denaturing the protein molecules that make up your hair, and that enables you to reshape your hairs from being straight into being wavy, or curly into straight, or whatever it is that you're doing. So those are examples of denaturing proteins. And the shape of the protein depends on the order you put the amino acids in. And there's an extremely important class of proteins called enzymes. And we will probably not finish enzymes. We've got about 20 minutes to go. but I will at least start on what enzymes do. All right, we have to look at metabolism a little bit more. Again, I'm not going to go into what chemical bonds are, these, mystical popsicle sticks that hold atoms together in molecules. We don't need to worry about what they are. Let's just say that in these chemical bonds, that's where we store energy. And energy can be released when you break chemical bonds, but energy is needed to form chemical bonds in the first place. Oh, the question is, how does cooking an egg change the order of its amino acids? The answer is it doesn't. What cooking an egg does is change the shape of the protein. The chain of amino acids is still chained together in exactly the same order, but the heat causes that folded protein to maybe unfold or fold in a different direction, or maybe some of the alpha helices stop coiling, or the beta sheets unravel, or something like that. The order of amino acids doesn't change. What changes is the folding of the entire protein. Proteins will fold into different shapes under different environments. So take a protein and put it in a hot environment like your frying pan, and the thing will refold in a completely different shape, which also changes the properties of the properties of the entire thing, changes the properties of the egg white or the properties of your hair. But it doesn't really change the order of amino acids unless you start breaking the protein down completely, and that's called burning it. So if you burned your egg white, you would. But with denaturing, you're not changing the order. Yeah, exactly. It's not the amino acids that are changing order. The entire protein changes its shape. Good question. Thank you. Okay, right. When you break chemical bonds, you release energy, but to form chemical bonds in the first place takes energy. And a chemical reaction involves breaking and releasing chemical bonds. Now, if that reaction ends up producing more energy than it consumes, we call the reaction exothermic, because it gives off the extra energy, often in the form of heat. And the reaction, once you start it, is going to keep going until everything is used up. And the simplest example of this would be fire. When you burn gasoline, or wood or anything, when you burn something, the thing that you're burning is made up of atoms that are stuck together in molecules. Octane, for example, the main component of gasoline, consists of 8 carbons that are stuck to 18 hydrogens in a particular pattern. When you burn gasoline, what you're doing is breaking most of those chemical bonds and reforming them, and you're adding oxygen to the blaze. That's why your car has an air intake. And by breaking all of those bonds that are present in the gasoline and forming new chemical bonds with the oxygen that's in your engine as well, you end up making carbon dioxide gas and water vapor and releasing lots of energy. Energy that your engine can harness to drive your car, or energy that gets just released as light and heat if instead of driving your car, you've just soaked your neighbor's house in gasoline and set a match to it. Not that I ever did that they could prove. But in both cases, you're giving off energy because you're breaking those bonds that held the gasoline molecules together and reforming new bonds with oxygen. and the amount of energy that's released is greater than the amount of energy that it takes to form those new bonds. And so gasoline will burn until there's nothing left to burn. That's an exothermic reaction. If a reaction consumes more energy than it produces, the reaction has to have an energy source to proceed. Like I don't know, like the chemistry that you do when you cook food. Okay. The chemistry that you use when you barbecue meat. There we go. When you barbecue meat, you're taking proteins in the meat called collagen. Okay, you've heard of that. That's in the extracellular matrix. Brisket has lots of collagen in it, and it's really tough. But if you cook it nice and slowly, you break down the collagen, and you can make the meat really tender and delicious if you're good at it. But that breakdown of collagen doesn't happen on its own, right? It's not going to happen if you take your brisket out and just stare at it. You have to keep adding energy in the form of heat, in the form of the heat from the burning wood in your barbecue pit. And you have to keep adding heat over a long time if you want to get proper, low and slow, tender, mouth-watering, melt in your mouth barbecued brisket. That reaction of collagen breaking down is endothermic because it doesn't happen on its own. It's not going to just keep going spontaneously. You have to keep adding energy, and when you stop, the reaction comes to an end. So that would be a reaction that is endothermic. You have to keep adding energy to make it go. So we can graph the amount of energy that's available in a chemical system over time. Think about burning gasoline. Once you start it, it's going to keep going. But it doesn't start spontaneously. What you have to do is add a little boost of energy. In your car's engine, this is provided by the spark plugs. You have to provide a little kick in order to get it started. If you're burning down your neighbor's house, you have to start it by lighting a match. It's not going to burn spontaneously. You have to give it some input, and that input is called the activation energy. And so we can draw a graph showing the amount of energy available in a chemical system over time. We might start with the energy being here, kind of on a plateau, and we have to add a boost. Once we have added that boost, the energy that's available is going to get released, and we end up with products that store little energy because all of that energy has been released. So we start with something like, in this case, it's sugar, which is the basic fuel for metabolism, but it could be gasoline. Those chemical bonds contain a lot of energy. To get at that energy, we have to give it our little boost called activation energy. And once we've given it that activation energy, the molecules start releasing the energy that they've got, and they end up as products that contain less energy in their chemical bonds because they've given up all that energy they were storing. Anything that lowers the activation energy of a chemical reaction is called a catalyst. One example of a catalyst that you might know, if you're driving a car made after 1974, which I assume that most of you are, your car's got a catalytic converter on the exhaust manifold. And the catalytic converter contains very fine platinum wires. That platinum acts as a catalyst. It takes harmful gases in your exhaust, like carbon monoxide and nitrous oxides and it turns them into forms that are less dangerous, like carbon dioxide, for example, which is still not great for you, but it's much less poisonous than carbon monoxide is. So the exhaust coming out of your car is cleaner than it would be. That platinum in your catalytic converter is the catalyst. And the thing about catalysts is they're not burned up by the chemical reaction. You don't have to keep adding platinum to your car, which is a good thing because it's really expensive. But that platinum is not burned up by all of the chemistry that it does. That platinum can last for the entire lifetime of your car because it's a catalyst, and catalysts don't get used up in chemical reactions. A protein that acts as a catalyst in a living system is an enzyme. Enzymes cause chemical reactions in your metabolism to happen by catalyzing them. Enzymes make chemical reactions happen because they act as catalysts for them. Incidentally, you have small organic molecules that are not proteins, but that play various helping roles in making chemical reactions happen. We call those coenzymes. And the only reason I'm bringing this up is that a number of vitamins are coenzymes. Your B vitamins, for example. What makes them good for you is that they act as coenzymes. You could think of them as enzymes' little helpers. And that's ultimately why you need them in your diet. Right. So if an enzyme is present, there is much less activation energy that's needed to get the reaction started. Imagine you've got a hill, and at the top of the hill, you've got a ball. Once the ball starts rolling down the hill, it'll keep rolling until it's at the bottom, right? It'll keep rolling until it eventually loses energy due to friction or due to running into something, right? Once the ball starts rolling, it'll keep going. Imagine, though, that there is a bump at the top of the hill, maybe a speed bump, that's holding the ball in place and keeping it from rolling down. If there's an enzyme present, what that does is take away the bump. Once that bump is taken away, now the ball can spontaneously start rolling down, even if there's just a little breeze that pushes it. It takes much less energy to get that ball starting to roll down the hill. And once it starts, it'll keep going all the way to the bottom. Activation energy and enzymes works the same way. Enzymes lower the activation energy needed to make a particular chemical reaction happen. And this is where this ties in with protein shape. The molecule that an enzyme acts on is called the enzyme's substrate. Quick example, one that you could try. You've got enzymes in your saliva called alpha amylase. And what they do is they start breaking down starches. If you eat starchy food, you start digesting it in your mouth. So if you were to take a saltine cracker and stick it in your mouth, at first it would be solid, but it would start breaking down. Now, some of that is because it would just get wet, but some of that is because you have alpha amylase enzymes in your saliva. And as that cracker gets soaked, the alpha amylase enzymes bind to the starch in the cracker, and they start breaking those starch molecules apart. And so the cracker gets soggy and limp, but it also starts getting more tasty. That starch in this case would be the substrate of your salivary enzymes. And the reason we think this works is that an enzyme molecule has a region on it called the active site, and the substrate fits into the active site much like a key fitting into a lock. or I don't know, much like a bolt fitting into a hex wrench. Once the substrate molecule is bound to the active site, the enzyme molecule might put stress on the molecule. It might stick it next to another molecule, depends. But the substrate molecule will end up either broken apart or possibly joined together. So there's a diagram of how this works. The substrate is shown as this funny shape in green, and the substrate fits into the active site of the enzyme. Now, once the substrate is fit into the active site, the enzyme might change shape a little bit, and the enzyme will put stress on the substrate and cause the substrate to break apart. So there at the very end, you can see those two separate products that have fallen out of the active site. And hey, now the enzyme is available for another molecule of the substrate to come in. So in this case, we have an enzyme that has broken down a substrate, but enzymes can also build substrates as well as break down. It depends on what enzyme you're talking about. All right, you could compare an enzyme to a tool like a socket wrench. The socket of the wrench would be the active site, and only one size or shape of nut or bolt can fit into the socket. So in this case, the nut or the bolt would be the substrate. The socket wrench, the whole thing would be the enzyme, and that enzyme's active site would be the socket on the wrench. So here's an actual model of alpha amylase. That's an enzyme in your saliva. And in yellow, that is a fragment of a starch molecule that's fit into this groove area. That's the active site. And again, when you put starchy foods into your mouth, the enzymes flood in. They're always present in your saliva. And the starches in your mouth make like Madonna did back in about 1984, I think, because they get into the groove. Or do any of you remember that song? Boy, you've got to prove your love to me. Yeah, only when I'm dancing. Okay, sorry. Right, anyway, so that yellow thing right there is a piece of a starch molecule that has fit into this groove. And once it fits into the groove, the enzyme can apply some stress that breaks the starch apart into simpler sugars. One more real example, this is called beta-lactamase. And you should be able to recognize by now it's got helices and sheets. And this little thing right here that's made of yellow sticks is a molecule of penicillin. When the penicillin fits into the active site, beta-lactamase causes it to break apart. You don't make beta-lactamase, but there are bacteria that can. And bacteria that can make beta-lactamase can break down penicillin, and that's how they resist it. This is why so many bacteria these days are antibiotic resistant, is they can make enzymes that break the antibiotics down. And in some cases, even use them for food. There are bacteria that eat antibiotics. And this is how they do it. They're able to make enzymes with an active site that fits penicillin like a lock fitting a key. Okay, that's actually a pretty good place to stop. And I hope I've impressed you with the importance of enzymes in living systems. So I will now stop the recording.